So thanks, Matteo, and for the kind invitations. It's very nice to be here. I will switch gear from like team talk, and I will basically talk about the impact of immune system on some kind of liver disease. In particular, I will focus my 15 minutes talking on colon cancer liver metastasis. That, as you know, is like the highest tumor burden for like liver diseases. Indeed, the colorectal cancer is the third most one worldwide, the second cause of cancer-related death, as you can see here. And then more than 50% of patients with colorectal cancer then will develop metastases in the liver. So surgical resections of the metastasis is the only options for potential cure of this disease, as you can know, and so we really need new therapies and, more important, also prognostic marker to predict the natural history of the disease. What is like the role of immune system in this regard? You know that liver is like the parenchymal organ, the solid organ with the highest infiltrations of immune cells. And the immune system is a major contribution factor in the prognosis of like colon liver metastasis patients as well of other diseases. Lymphocytes contribute to liver homeostasis and tumor immune surveillance, and interactions between tumor cells and tissue resident or infiltrated immune cells is extremely important for the natural history of the disease. We already demonstrated with Matteo and other colleagues at Humanitas University of Milano how important is, like, for example, the frequencies of NK cells or T cells in natural history and prognosis of liver metastasis from colorectal cancer. Today, I will focus the presentations and to give like a case of study also for unconventional T cells called gamma delta T cells. As you know, like the T cells word is divided like in the conventional alpha beta T cells and then most recently also of unconventional gamma delta T cells. What are like the difference of these T cells compared to conventional T cells? Basically, they are like much more frequent, like in peripheral blood, but they are only enriched in tissues, as you can see, like in these tables, and they present any kind of antigens, including phosphoantigens, without restrictions of class 1 presentations. So they are like a perfect compound of cells, extremely cytotoxic against the tumor, at the border between innate and adaptive immune system. The relevance and the knowledge about the important role of gamma delta T cells in tumors is not that old. It only started 20 years ago with these first studies like on skin cancer in Science in 2001 and then in other tumors. But then a seminal study came out like in 2015 on natural genetics, on natural medicine, in which among all the immune cells participating like in the physiopathology of cancer, it was demonstrated that gamma delta T cells and high frequencies of gamma delta T cells represent the most favorable immune parameters for like predicting a good prognosis and natural history of the diseases of the cancer. So gamma delta T cells have quite a lot of functions, but today we'll focus, of course, on their anti-tumor activity. They are performed by two main subsets. I will try to make this story very simple. On the VδT T cell subset, which is basically mainly present in peripheral blood, and Vδ1, which is mainly enriched in tissues. Both of these subsets has a very high anti-tumor activities, and they are used today for several adoptive cell transfer protocols for a lot of tumors. Let's see what are its impact in colorectal liver metastasis here. Indeed, there are many challenges, like in gamma delta world, for protecting against tumors, and they relies again on the diversity of the T cell subset. The Vδ1 in tissues, as you can see here, the Vδ2 in peripheral blood, and the different repertoire they have in the TCR basically perform a different tumor reactivity against different kinds of tumor. Today, I will focus a little bit on Vδ1 T cells and why they're important. Our lab already demonstrated that like these Vδ1 T cells are extremely important, both against tumors, against viral infections, but also against like tumor of the gut and of liver, as you can see here. But they are very high infiltrating the liver, as you can see here. Indeed, unconventional T cell represent one of the major T cell subsets present in the liver. And so what we wanted to do is to understand the impact in the natural history of colon liver cell metastasis. So what, in collaborations with Matteo at Humanitas Research Hospital, we basically enrolled several patients with primary CRC. As you know, these patients underwent surgery plus chemotherapy, but 50% of those patients developed 1 year later, at least 1 year later, colon liver metastasis. Some of these patients only 20% went directly to surgery, while 80% underwent neoadjuvant chemotherapy. For those that went anyway and the tumor progressions, a second cycle of chemotherapy were performed in order to get a tumor stability, a tumor regression in these patients, and they were under surgery. In this case, basically, we recruit these two cohort of patients, and we analyze the role of immune system, impact immune system on natural history of diseases. Basically, we took samples from peripheral blood for the tumor and for intratumor of these cells. We digested all the specimens, and we obtained suspensions of single cells in order to study several parameters and impact of immune receptors in the history of the diseases. As you can see here, this is an example of how we obtain the cells within viable lymphocytes. As you can see here, within the CD3 T cells compartment, we got the VδT1 and VδT2. So what we saw? We saw first that in the peripheral blood of patients with the CLM, there is a decreased frequencies of CD3 of T cells overall, a decreased frequencies of the VδT2 compartment, but a stable presence of VδT1, which in terms means that there is a relative increase in the peripheral blood of CLM patients of cells having the VδT1 T cell compartment of the cells. Now, in order to understand which kind of cell is that, and again, I will try to make the story very simple, just remember these two markers, CD27 and 45 array. They basically, the combination of that, they can identify some subset. And I would like you to remember about this subset called TEMRA, which will represent the cell subset with the highest potentials against the tumor cell target. So the TEMRA differentiated cells that are able basically to kill tumors, which are basically highlighted here in light brown, as you can see in this diagram. Now, if the presence of this high anti-tumor effector is among 50% under homeostatic conditions, in the peripheral blood of CLM patients, basically they are highly increased. You can see up to 70%, which means that these patients have circulating cells with high capability of killing the tumors. If we go back to the clinical features of the cohort of 93 patients that we enrolled, there is one feature I would like to drag your attention on, which is the fact that 70% of these patients underwent neoadjuvant chemotherapy, as I basically told you at the very beginning, while only 20% of the recruited patients were naive for chemotherapy. Now, we want to understand whether the chemotherapy had any effect in the expansions of these cells. And surprisingly, we found either in the absence of chemotherapy or in the presence of chemotherapy, we still had a very high expansions of this cell subset able basically to kill the tumors. So there is an increase of these VδT1 effector memory cells that is completely independent and resistant to the chemotherapy, which is a good thing, and I'll explain you after. Then basically we found that these cells circulating in the blood of CLM patients have also a very high expressions of a marker called the CD69. Why it's important, CD69, among all other markers? Because this is a constitutive markers of tissue-resident immune cells, including cells present in the liver, so hepatic lymphocytes. And the expressions of this high expressions of this marker, CD69, also correlate with expressions of other important activating anti-tumor receptor like CD61 and CD56, and many others of these. Now, if we see now the distributions of these cells among the peripheral blood of healthy donors in green or CLM patients in yellow, you see that the distributions of subset is very different in these two cohort of healthy donors, the green, and CLM patients in orange, as you can see here. And this distribution is still more specific in we restrict the distributions to the terminal effector area cells, as you can see here, but became almost specific only of CLM patients when basically we only looked at those cells expressing a tissue residence marker CD69, as you can see here. So what is the take-home message? Is that like in the blood of like these patients, CD69 identify specific populations of like effector memory T cells that cluster only in the blood of these patients and not in healthy donor. So we found the populations of cells that seems to be specific presently at high frequencies like in the blood of CLP patients. But where these cells come from? So we then analyze also the liver specimens of the same patients. This is like, as you can see, one of the specimens that Matteo gave us after surgery. And you can see that like there's an high infiltrations of cells just around the tumor, as you can see here. And most of these cells were just the gamma delta T cells, as you can see here, that, as you can see in this diagram, basically focalized around the tumors, around the peritumor areas. And most of those cells are, again, cells that are VδT1 positive, so specific of the liver, specific of the tissues, in this case of the liver. Now, these cells, again, were within the different compartment of the cell's response, mainly effector memory. So cells thermally differentiated with high anti-tumor potentials, as you can see here. And again, the high expressions of these cells correlated with high expressions of activating and anti-tumor targets, again. So as you can see here, this is like the case of the liver. Now, if we compare the cells from the one of PBMC, they are exactly the same feature, the same phenotype. And the higher are the cells in the liver, the higher are the cells in peripheral blood. So there's a direct correlation of the frequencies of this these cells into compartments in the liver of PBMC. So what is like our working hypothesis? That could be like a dynamic exchange of tissue-resident cells that from the liver around the tumor could extravasate in peripheral blood of the same patients. And to understand this, we did like quite a few proof of concept experiments, including the sequences of TCR that could basically track at clonal level every single cell extravasating from the liver to the blood, as you can see here. And in particular, we sequencing like all the cells from the liver for TCR, and then the cells expressing CD69 or not expressing CD69 in peripheral blood, as you can see here. And what we found that among like the clones of cells shared by the liver and peripheral blood, there is like a very high significant number of clones among the cells expressing the CD69 positive, as you can see here, so tissue resident, while only a minor number of clones were shared among the CD69 negative compartment, so not tissue resident cells. So what is the take-home message on this? Is that a significant rate of CD69 positive and tissue resident cells expanded like in the liver of CL patients can extravasate in peripheral blood. They are exactly the same cells that we can follow a clonal. And to long story short, we had also another like series of like proof that confirmed like the same results, as you can see here. Now, what is the clinical impact of these cells extravasating from the liver, from the tumor to the blood? As you can see here, we first found that like high is the frequencies, like both in the liver and peripheral blood, the low number of metastatic lesions in the liver. And as you well know, like the presence of less than 4 metastases in the liver correlates with like a good or better prognosis of patients. Then what we also found, we found that basically the higher was the presence of these cells within the lesions in the liver, the longer was overall survival of the cells, as you can see here in both graphs. I just show you that some of these cells basically could extravasate also in the blood of the same patients. And again, even like the higher frequencies of the cells, not only in the liver, but also in the blood, correlated with a fantastic higher, overall significant higher overall survival of these patients, as you can see here. So higher frequencies of these cells, both in the liver and peripheral blood, could predict a better and longer natural history of like the disease, as you can see here. To finish, I would like also to highlight another concept, which is the following. Is it possible to manipulate in vitro these cells and propose these cells for adoptive cell transfer therapy to improve the anti-tumor response to this, like against these tumors? So to do this, we try some experiments that we are still performing now, in which we culture directly the cells from the blood of these patients with the autologous cells that we derived from the liver specimens that Matteo gave us. So we put the cells in co-culture, basically, cells from the liver, T cells, plus the tumor cells from the liver of these patients, and we put them in cultures. So as you can see, at day 0, we put like several clones of T cells, quite a few, as expected. We put like the entire totality of delta 1 from the blood of these patients. But after 14 days, we get like in 3 out of the patients we tested, like monoclonal expansions of a single and very efficient anti-tumor clones of these cells, as you can see here. And this was obtained in 3 out of 8 patients that we tested. So as the tumors in vitro, and then it's possible to reinfuse these cells for adoptive cell transfer therapy after. To improve the natural history of like the disease, as you can see here. So in summary, to conclude, what we tried like demonstrated to convince you about, that like during CLM, there is like an expansion of cells in the liver tumor around the tumors that express marker of tissue resiliency, as you can see here. These cells, basically, they can recirculate here through like the blood of like the patients, and it's possible to identify these cells also like in the peripheral blood of the same patients. And this like identification is absolutely independent from chemotherapy. So whether the patients do or not neoadjuvant chemotherapy, we see like these cells. The cells highly correlate with the natural history of the museum of the lesions of the cancer. So the higher is the frequency, the longer is the overall survival of these patients. And there is also implications for using these cells for adoptive cell transfer immunotherapies, because it's possible to expand the cells in vitro and then infuse like in the same patients after a few weeks. So this is basically was just published like a couple of months ago in this paper, where you can find like all the details about it. And having said that, I would like to thank like all the people that collaborated with these studies, especially like the group of Matteo and all like our collaborators in my group, especially Ioanna Mikulenk and Valentina Cazzetta. And I would like also to thank everyone for your attention.
Thank you very much, Domenico, for this very interesting presentation. I think we have done a very good job together in the last years. Let me see if there is any question from the audience.
Yeah, first of all, thank you very much and congratulations for your cooperation. I have 2 short questions. The first one is, could it be that, let's say, the tumors, they have different KRAS mutations, so they may produce different chemokines, and you have, just because of that, more CD69 around the tumor? And the second thing is, you know, I am quite old, and 30 years ago, Alex Knut and Diepold did the same thing with melanomas. I realized working together with them that it's quite difficult to get tumor cells in culture. These are the two different questions. So the first thing is, do you have a KRAS analysis or mutational analysis of the tumors of the metastasis?
So we were able basically so far for this study to recruit 93 patients, but we have much more. And so far in our experience, we didn't see any difference either in the presence or the absence of KRAS mutations. So the expansions of these cells, as well as independent from chemotherapy, was also independent from like the main mutations, including KRAS mutations we got like in our 250 patients that we recruited over like the past 3 years with Matteo. As for the second questions, it's very difficult to get in culture tumor cells. You are extremely right. But like the technology now improved a lot, and you can even like buy cell lines or like tumor cells, primary cells, which basically try to resemble in vitro like the niche that you have for tumors. So the answer is yes, it's very difficult, but technology now allows us basically to get much better results even compared to 5 or 10 years ago. And for the liver, basically we get like, 8 patients for this to try to expand the cells from 8 patients to put them in culture, and we only were succeeded with 3 patients. So we have for sure to study much more to understand like how to increase the efficiency of the system. But to make a long story short, now it's like possible, but of course it must be improved with technology.
Good luck.
Thank you.
A comment and perhaps a suggestion to Domenico from our group at Imperial, it might be the possibility of looking at organoids, and there is the possibility of potentially interacting from the angle, so to sort of maintain the microenvironment architecture over time. And there is another really interesting technology, which is precision cut liver slices, that is particularly taking off at King's College Hospital. They've also had an ESOL school specifically looking at how to maintain in culture a living slice of tissue for up to 14 days, where you can actually look at the dynamic changes in the immune population. I think those models for you guys would be absolutely fantastic to look at the dynamic changes in this T cell subtypes, in particular T cells.
Thank you, David. There was two very nice suggestions. We're already working with organoids, not only like for liver, of course, but organoids are very popular now, but also the second technique with like liver tissue space. So there are two very interesting like technologies that we can follow over the years. So we'll try like to increase our efficiency for sure.
Hi, Domenico. This is a really nice presentation. Congratulations for your story. It's really interesting also for the further application. I have just a curiosity because you show that your cells, your gamma delta 1 subtype is the perimetastatic area. I was wondering if you also have a look inside of the tumor, and if you have an answer, because those cells need to be expanded from the tissue and go into the periphery, because actually in the peripheral blood, what we can expect to find disseminating cancer cells, but if you have already the metastasis in the liver, what will be the target of those cells?
Okay, so we didn't find surprisingly, I don't know how much surprise it was, but we didn't find many cells inside the tumor. So within like the tumor, we found a very low frequencies of the cells, but the cells were only very highly enriched around the peritumoral area. Like, they were trying to get into the tumor, but they were not able like to get into the tumor. So now about the targets, we also study like the same compartments in patients doing at the same time both surgery from the colon and from the liver, in order to see if the cells have the same targets or different targets, and whether like they could protect from the progressions of the disease. So we don't know which is the specific target of these cells, but now we are trying basically like to expand our project to see whether like this is a specific target going from the colon to the liver, or whether this is like a tissue specific phenomenon. We don't know yet that. So
you don't know actually what those cells do in the tissue. So what is the target of the department?
Yeah, it's very difficult. It's very difficult to try to understand the specificity of these cells versus like, tumor antigens or versus like a cell antigens. So
also
since
because
it's like a disease,
you are super enriched of other immune population
of course,
in that area. So it's
of course.
completely,
but since we have now the possibility of testing the cells from both sides of the same tumor, both from the liver and from the colon, there will be, we are doing like a very complex experiment of single cell sequencing, as we did also for this study. But for the sake of time, I didn't have the time to show everything up, to understand whether like these expansions, together with the sequencing of TCR, it's possible to make to give us a better knowledge about the specificity against the tumor antigens or not. And so we are working on it.
Good luck. It's a super interesting work. Congratulations.
Grazie. Thank you very much.
There is another question, I think.
Please. I cannot hear the question.
Mr. Moad Atsam.
Can I ask question? about the microbiome. What is the role of microbiome in the treatment of cancer in this situation?
I don't know. We didn't do like in experiments of 16R, so on like both like in the gut or in the liver. So whether like the microbiome has an influence, like in enriching or expanding the cells, simply I don't know. So sorry if I cannot give like of any more informations, but we might make the experiment.
Okay, thank you very much, Domenico, for this very brilliant presentation.
Thanks for having me.